Hi, and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at seed germination, which is part of your plant response module. So what is seed germination? Seed germination is when a seed starts to grow and the root and the shoot will actually break out of the seed coat, and what we will see is the shoot will grow one way and the root will grow another. Well, what causes this seed to germinate? Well, that will only happen if the seed has optimal conditions. And one of those conditions that it requires is water. If we water our seeds, what will happen is the seed will absorb water, and that will make the cells become metabolically active. What then happens is those metabolically active seeds will start to produce gibberellins. Now, gibberellins are the hormones that are produced within the cotyledons, which are a part of the dicot. So the dicotyledonous plants produce these seeds that have dicots, two cotyledons, and the cotyledons are the ones that produce the hormone gibberellins. Now this gibberellin is going to switch on genes. That will mean that transcription and translation will be able to occur. Now that stimulating of the genes being switched on causes the production of proteases, an enzyme that's going to break down proteins into amino acids, and then those amino acids can then be used to produce amylase, another enzyme. Now, what do we know about amylase? Amylase is an enzyme that is needed to digest starch. So the amylase is necessary to hydrolyze down the polysaccharide, the starch. So this is where your module 2 biological molecules knowledge needs to come in. We know that starch is made up of amylose and amylopectin. Starch is a storage molecule within a plant. So the seed has stored this glucose in the form of starch. Well, it then needs to hydrolyze that storage down to release the glucose. But why? Why does it need to release the glucose? Well, that glucose is needed for respiration. The glucose will be broken down during glycolysis, and then with enough oxygen, aerobic respiration will occur. Now, aerobic respiration means that there will be a higher production of ATP molecules. So therefore, another condition for seed germination is about this availability of oxygen and water. We were talking about enzymes, so therefore we need to ensure that the temperature is correct. So if we were going to do an investigation on this, these are the types of variables we would want to control. So this is where they could link seed germination and plant response to the module 5 respiration module. But remember, actually, for plants to grow, they need to not only be provided with the ATP molecules, but they also need to synthesize cellulose to make the cell walls for their growing cells. So therefore, we could use that glucose that was stored as starch and convert it into the cellulose to create that cell wall. Or it might be used to actually produce things like your amino acids or lipids, depending on what the plant actually needs for the growth. So it's important that we note for seed germination that gibberellins are really important. But what we also have to understand is actually there is evidence that shows us that the other hormones which the plant will produce, for example, abscisic acid, actually works antagonistically with the gibberellins. So to maintain the seed dormancy until those conditions are right, that is because of the hormone abscisic acid. When the hormone levels decrease with abscisic acid and increase enough with gibberellins, that is when the seed will actually go through that germination. So we know that abscisic acid is about maintaining seed and bud dormancy, and therefore it is a very fine balancing act, and all these hormones actually work together. So how do we know this? Well, we know this because when they actually took mutant variants of seeds which were not able to produce gibberellins, they did not germinate. When they externally applied the gibberellins, the seed then germinated. So that proved that gibberellins was the hormone that was necessary for seed germination. And if they were taking a seed and they applied inhibitors, i.e. abscisic acid in a higher concentration, once they removed that and got that level down to a low enough concentration, then the seed was able to germinate. So by doing investigations, we can actually see how these hormones interact. So I hope you've liked this video, and if you have liked it, then please do click on the like button and subscribe to my channel. Also, if you haven't done so already, do check out my revision platform, www.aiqchat.com.